I'm Monica with Nucleic Verse. I use analogies, metaphors, and anthropomorphization in order to create poems and story. It's how I understand. It's how I see the unseen and ponder the unknown. Just as words are imperfect models of thought, so too are these words strung together as story. But imperfect as they are, we try. In this poem, I describe my relationship with samples I tested for a genetic alteration. The samples were usually from breast cancer and most usually from women. I was able to view cells taken from someone and describe what was happening in a representation of their tumor, while somewhere outside the lab walls they waited for the results to inform their treatment. Her2. I see you. I know what you did. You forgot your place, your neighbor's fences, broke free from unspoken restraint. And what did you earn? Expand and live forever? How long had these plans been hatching? Did you even let her know? You didn't ask. The stage is all yours now. Tell me how you did it. Icarus, embedded in paraffin, 7 microns deep. Hubris to want to live forever. It can't be done. Fresh, frozen, now glowing red and green, revealing your deed. You didn't make it. You took her too. Now let me give you a little bit of background to help explain my word choice of the poem. Your entire body works because of cells. Your body systems and organs are made up of cells, loads of different kinds and shapes, cells that are packed into groups to form tissues and organs, and they need to know when to stop growing. This is called contact inhibition. Cells generally respect the boundaries of neighboring cells and do not divide too fast as to crowd out their neighbors' nutrition and function. There is a need to grow faster than surrounding cells' tissues during such times as embryonic development, puberty, wound healing. But there are scads of signals and hormones to direct this growth in a highly controlled, highly coordinated way. Grow here, not here. Slow down. Stop. Start again. But sometimes the cell overreacts to a signal, or the call is coming from inside the cell itself. This unregulated, unguided cell division can create a group of daughter cells that can divide faster than their neighbors. This creates a lump and is the result of proliferation beyond control. This doesn't necessarily cause a problem. Many more mutations and disruptions are required for a rogue group of cells to disrupt the overall health and function of an organism. There are many steps toward a cancer developing. This loss of contact inhibition is just one of them. The poem then talks about wanting to live forever. This refers to losing sight of a very beautiful process of taking turns between the cells, known as programmed cell death. Now, that doesn't sound so beautiful, but hold on. It is so necessary for our existence and so poetic. I'll dedicate at least another dozen or so poems to it. Cells are not forever. There are signals that indicate the time of their usefulness has passed, and some cells are very short-lived. When this happens, the cell packages up their contents into little bubbles, and then the overall outer membrane disintegrates, and these bubbles are absorbed and taken in by the surrounding cells. Massive recycling, if you will. This is cell turnover, and it is absolutely required for tissues, organs, and an organism to replenish cells that are past prime with new cells full of vigor. Mind you, the new cells are the daughters of the old, but that's a story for a different poem. So, expand and live forever refers to proliferation beyond control, loss of contact inhibition, and dysregulation of programmed cell death. When I pose the question, how long have these plans been hatching? I'm wondering when the mutations accumulated. Was there a genetic predisposition? Did it happen over decades or a few years? The story of each cancer is very unique. We will not know just by looking at a tissue under the microscope when the mutations happened or how. We can make guesses based on experiments, and someday maybe we'll be better able to read the time-stamped ticker tape regions of DNA and be able to better piece together this story. I wanted to pay homage to the life of the individual whose tumor was now before me, just imagining for a moment what was happening in their life when these cells changed in a way that would change their lives in such a devastating way. The microscope stage is a flat surface. I placed the slide to be examined on it. It seemed fitting that it was named stage, as the slide had all my attention. In order for me to do my analysis, a piece of the tumor was immediately preserved, either by freezing or chemicals, and then sliced into extremely thin sections. This is what I'm referring to when I say fresh frozen, the preservation method, so that the DNA within the cells doesn't break down before I have a chance to see it under my microscope. Green and red are the most common fluorescent colors used in a technique called fluorescence in situ hybridization, or FISH. FISH microscopy is beautiful and so cool. To explain it simply, we use colors to see gene regions, but gene regions are way smaller than microscopes. So, how is this possible? Well, the tissue is flooded with probes, sticky molecules that have a particular and specific attraction for the genes we're interested in. On the other side is a fluorophore, a fluorescent signal that is powerful enough to see under a regular microscope. We used red to stick to the chromosome region named Her2, normally found on chromosome 17. And we used green to stick to the centromere of chromosome 17. Humans with a genetic makeup compatible with life have 2 of every chromosome. There are some exceptions, a story for a different time. Each gene shows up once on every chromosome. So in a normal cell, there should be 2 red and 2 green fluorescent glowing regions. But wait, what happened here? We have the number of chromosomes represented by the green regions, but there are so many more red signals. What we are seeing is evidence of gene amplification of the Her2 gene region. Remember before, when I said that the cell can proliferate beyond control if it overreacts? Well, this is potentially one of the reasons why the tumor we see here came to be under the microscope. The Her2 gene codes for a protein that acts as an ear to listen for a signal to divide. Her2 sticks in the cell membrane, and when activated, it starts a cascade like a Rube Goldberg machine to start the cell division process. What if you have a ton of Her2 genes that make a ton of Her2 proteins that stick in the membrane? A cell covered in ears. Even a whisper or rumors of cell division starts the cascade. So many hours I spent scrolling overlapping fields of view through the microscope, seeing the lights of Christmas signifying something very different. It was beautiful, sad, and hopeful all at once. Her2 amplified tumors respond to a medication that specifically targets the protein. It sticks wax in the ear, so to speak. This has greatly improved the treatment and outcome of these cancers. But cancer is a shifty, moving target, more able to gather mutations now that some of the regulations and controls have been removed. I was thinking about the story of Icarus, the boy in Greek mythology who escaped imprisonment with his father with wings crafted from feathers and wax. Not listening to warnings, Icarus flew too close to the sun. The wax melted, and he fell back to earth and died. We use wax to coat the piece of tumor. This allows the blade cutting the tissue to have even pressure applied and get a nice, clean cut. We also use warm slides to attract the thin sections. They almost jump up onto the slides. So I thought it was apt that the tissue that attempted to defy natural constraints be embedded in wax. The phrase, fly too close to the sun, is used to describe ventures that are too lofty. And so it seems that Icarus and cancer have some things in common. The desire to do more, conquer, is all over literature and the media. We want to live forever. We want to break free from constraints. We want to forget our neighbors. And yet, when our cells do so with a precise set of characteristics, the outcome is cancer. Thus, the Icarus reference in the poem.